Okay, hi, my name's Tamara and I'm a research assistant from the Blewett Lab at the Walter and Eliza Hall Institute. In our lab, we study a large epigenetic repressor called SMooCD1. SMooCD1 is involved in X chromosome inactivation, and it's also involved in the silencing of clustered gene families like Hox genes, protocoherins, and imprinted gene clusters like the Prader-Willi locus on chromosome 15. A potential therapeutic approach that we are investigating to treat Prader-Willi syndrome postnatally involves depleting this SMooCD1 protein and seeing if we can activate gene expression from the maternal alleles that it is silencing. Our early experiments in human epithelial cell lines have shown that when SMooCD1 is knocked out, Prader-Willi genes MAGEL2 and McCoran3 are significantly upregulated. And earlier today, Megan Aminatov from our lab presented her promising in vivo work in our MAGEL2 mouse models, showing the activation of the maternal copies of Prader-Willi genes in the brain when SMooCD1 is depleted embryonically. So we're now focusing on repeating this work in more relevant human cell lines. And we've turned our efforts towards using induced pluripotent stem cells, or iPSCs, that are generated from different Prader-Willi patient lines and healthy controls. We are differentiating these iPSCs into neural progenitor cells, or NPCs. NPCs express Prader-Willi genes, and they also proliferate and can differentiate in culture. We're hoping that by working with this cell type that we can better study the effect of SMooCD1 loss at a stage that more closely resembles the postnatal therapeutic window that we're hoping to target. In order to deplete SMooCD1 in NPCs, we're using antisense oligonucleotides, and these are diluted directly into the cell culture medium where they enter the cell and bind to the messenger RNA of SMooCD1, causing exon skipping and then SMooCD1 protein depletion. Our early results from both 4-day and 7-day knockdown time courses are promising, and with gene expression analysis by RT-qPCR and by bulk RNA-seq have shown upregulation of the Prader-Willi genes MAGEL2 and McCoran3 and SNRPN from the maternally imprinted cluster. And we also see little to no change in nearby non-imprinted genes or UBE3A, the repression of which may cause Angelman syndrome. So we're currently in the process of replicating these experiments in other batches of NPCs and NPCs that have been differentiated from other Prader-Willi patient lines. And to date, our activation levels that we're seeing don't match the paternal levels from healthy controls. And so we're also currently investigating whether we can increase gene expression by targeting other factors that might also be involved in gene silencing. And we currently have preliminary work showing that the effect can be boosted in human epithelial cell lines. So please come and chat to me or come and see me at my poster. I'd love to talk to you about the work that we're doing. Thank you.